Good morning, everyone. Thank you. We're going to move into session 2, Pathogen-First, Phenotype-First, and Sequence-First approaches to genomic medicine implementation in infectious disease. And we're going to start off with our very first talk, which is from Dusan.
Hi, everyone. Thank you for invitation, and thank you for organizing this meeting. I'm enjoying it quite a bit. Do I have to be a little bit closer? Okay, I'll do that so you can hear me. Okay, so let's talk about rare infections and how we approach them currently in the clinic. How can we implement that, and maybe what are the challenges of things to come. So first, thank you to people who did a bunch of work in my lab. Inborn errors of immunity were described extremely well by Jennifer Puck here, but essentially we have about 500 genetic disorders that lead to them. Jennifer likes this graph if she pointed the other way around, and I don't disagree. But what this is a phenotype-first approach. So we start with somebody who we think needs sequencing, and then we do the sequencing, identify, and those that are luckier than others, sometimes there is actually precision medicine intervention that's absolutely wonderful to see. Okay, so mutations in these immune genes can lead to essentially susceptibility to infections via bone marrow failure, cellular deficiencies, or antibody deficiencies that we've touched upon, and also they can lead to autoinflammation, immune dysregulation, which is really this immune dysregulatory phenotype that people have mentioned. So as I was putting this talk together, I was like, well, what is the right way to implement this? And just at the time, Scott actually emailed me and said, hey, this is what we use, right? And here is the immunodeficiencysearch.com, which is an algorithm. This is something that any provider can use in their decision making, whether or not to sequence an individual. And clear, as you can see, if the patient is not responding to therapy or not clearing the infection, I would say no matter the age. Of course, in children, genetics is going to be more penetrant, but now we have quite a few cases of adult onset disease. You can click here on, let's say, viral disease, that this patient is not clearing, and it's just persistently there. And if you click on that, the next thing that can show up is this CMV, is this herpes, is this very severe influenza, is this JC virus, BK virus, HHV8, varicella, HPV. And here for just simple purposes, let's say we click that this patient has recurrent herpes that is not just labial, but is showing up on other parts of the skin, and what could be. So if you go and click there, this is what shows up. It can be NEMO deficiency, it can be, of course, Wiscolt-Altrich treatment, it can be STAT1 deficiency, TLR3 deficiency. So at that point, what a provider can do and should do is say, okay, so it could be genetic, let's send it for sequencing, right? And what do I do? How do I do that? Where do I send for sequencing? So Invitae panel, which we've talked about here, covers 429 genes currently that are known, mutations in which can cause some of these infectious and otherwise phenotypes. So results come back with a variant that is called pathogenic, case solved, meaning there is exact same variation in TLR3 that causes herpes simplex encephalitis. Let's say that's the case. And now we know that we have a genetic diagnosis and there is genetic counseling for that can be done for the family members. Most often what happens is that the results come back with a variant that is called unknown significance. So you have a VUS, or variant of unknown significance, and that's really the stumbling problem right now when the results come back to the provider. This is where the provider says, what do I do? Right, so they can email Ivan, they can email Scott, they can email me, and usually it works through that. There's a couple of listservs that providers may or may not know about, depending how intimately they're linked with the research community to seek help for that. So NIH has heard us and funded 2 different programs. One is New York Royal, the other one is NICER, which helps with this functional validation and ideally reclassification. Because for Ivan's team ever to look at this variant of unknown significance, it needs all these functional and all other white tests that need to be published for them to be part of the ClinGen, so then Invitae can call this pathogenic, as opposed to a variant of unknown significance, right? So there's really layers to that, and these two programs help accelerate the speed at which we can feed our data via publications and direct submission to ClinGen for this reclassification to happen, so that provider gets a result back and need not consult us on more things. So that's one idea. So results come back negative. What happens is that if you decided that it was worth to do a 429 gene panel, it's probably good to refer to research, whether it's a whole exome or whole genome sequencing research that can then again start the same process with us identifying novel variants or even novel genes that can cause these infectious diseases or immunodysregulatory syndromes. Okay. So how can NIH initiatives expand use and diagnosis of infectious diseases via whole genetics? Currently, most variants in genes remain functionally not annotated, and this is true not just for genes that cause infectious diseases, this is true for genes that cause any disease. So one way, mostly we have 2 to 3 known disease-causing variants. Most identified will be variants of unknown significance. So I'm of the thought that we need a forward-looking genomic database. It's a little insane, but we were insane before, and Whole Genome Project brought us to where we are today in medicine, which I think is absolutely unprecedented. So the next step in my book is to do a forward-looking genomic database where we functionally annotate every and single SNP in every and single gene, and go beyond that and functionally annotate at saturation every single nucleotide, so we can be forward-looking, so when somebody comes with a mutation, we already know functionally whether or not in vitro this is associated with a phenotype. This gets very complicated very fast, and it's probably beyond this meeting. This is just what I said. Okay. We had a Human Genome Project at the beginning of the century. I think it's time for the Human Variome Project for the next half of the century. That's probably bigger than the Genome Project. Okay. Another problem that Jennifer also alluded to is in the dominant disease especially, we have incomplete penetrance. Incomplete penetrance happens when there is a carrier of otherwise known disease-causing allele that is not sick, or partial disease expressivity, meaning it's not as sick as some other family members. And we have a very nice term for it, incomplete penetrance, but we don't understand it. Here, I'm just showing you the example of a made-up family, but everybody carries the allele, and you can see that the father and the daughter have fully clinical disease. There is a partial phenotype in a sister, whereas the mother, who's wild type, and the son, who is a carrier, are completely healthy. So this is a classic pedigree of incompletely penetrant family. If this was the gene first to be looked at, any geneticist would look and say, this is not the disease-causing gene, right? Because it just makes no sense. Okay. So what I had to do recently is go back to textbooks and relearn that actually not all genes are expressed from both alleles. And of course, we know this from genomic imprinting, where you can be just expressing maternal or paternal allele throughout your organism. We know, of course, of X chromosome inactivation. Individuals with 2 X chromosomes, one gets randomly inactivated. We know about transcriptional bursting, when one allele gets preferentially transcribed really fast, and that's been described by actually Bill Paul here at NIH in the 80s. And what I'm going to tell you a little bit about today is the concept of monoallelic expression, which is different from transcriptional burst because it's actually mitotically stable and it's somatic. So you can think about it as transcriptional mosaicism. So you're heterozygous everywhere, but let's say only your T cells become transcriptionally homozygous, and that's actually pretty stable. So that suggests that if we think about incomplete penetrance, canonically we would see somebody with partial disease expressivity, no disease, or fully penetrant disease as equally expressing maternal and paternal alleles. But if we twist this thinking a little bit, we can think about somebody who's healthy that's predominantly expressing the wild type allele. Somebody who's a little sicker has more of the mutant allele expressed, and somebody who's very sick, despite equal genomic heterozygosity, is actually expressing mostly the mutant allele. Okay? So how did we come about this? We identified a patient with a mutation in JAK1. This was a de novo mutation, and it was a mosaic mutation at DNA level. So what we did is we did single-cell RNA sequencing because within the individual, we had both wild-type cells and cells that carried the mutation. And here is the result, if you can look at there. The wild-type allele should be expressed in every cell because it's either wild-type or a heterozygous cell, and 50% of cells should have it. What was shocking to us is that the other 50% of cells should be double positive for the mutant and the wild type. And what we ended up seeing is mostly the mutant allele expressed, which didn't make any sense because it's against what we were taught in very basic human genetics, right? Okay, so we went on a quest for about 5 years and looked at, formed first hypothesis that monoallelic expression contributes to phenotypic variability or incomplete penetrance in inborn errors of immunity. And we tested this in 10 healthy New Yorkers, and let me orient you around this graph. On the top is a donor. There is donor A, B, C, D, and F, and columns are number of clones of T cells that we expanded from a single cell to 200,000 cells. If you look at the color of the circle, the middle is yellowish and light blue. That means it's biallelically expressed. If it's completely blue, it means it's an alternate allele. If it's completely red, it means it's a reference allele. If you go to donor C, you can see how about half the clones are monoallelically expressing a silent SNP that this healthy individual has. If you go to donor F and PLC gamma 2, you can see how one clone is completely maternal, one is completely paternal, and the rest of the clones are biallelic. So this suggested to us that this transcriptional mosaicism, or the ability to somatically commit to the expression more so of one allele, is actually a common trait of human biology. And long story short, we know now that about 5% of genes can undergo monoallelic expression. In each individual that we tested, N is small, but we've never detected more than 0.5% of genes undergoing monoallelic expression, at least in the immune system. Does this influence incomplete penetrance? Here is a family where the grandfather, mother, and the granddaughter all carry a variation. The mother is the only one that's sick. Again, classic genetics would say this is not the allele that can cause disease because you have two healthy individuals, older, younger, they're not presenting with a phenotype. This is an interesting case because this is a female from the UK. She was healthy until toxoplasmosis at the age of 28. Following that, she had recurrent infections, CVID, hyper-IgM, splenomegaly, severe cytopenia, thrombocytopenia requiring transfusion. It's very clear that toxoplasmosis doesn't cause this, but it correlated with it. So what we did here is with biochemical testing, something that we can send to Iman's group to evaluate with, and as you can see here, compared to the wild type, it's a very clear biochemical gain of function. There is no question about it. But the grandfather and the granddaughter also carry the allele with this very clear gain of function, and they're not ill. If you look at then what we did, the next thing is we sorted B cells, monocytes, NK cells, and T cells. The graph, the y-axis, is JAK1 mutant allele expression. If somebody's a heterozygous, all these dots should be around 50. And what we saw in the mother and the daughter, that predominantly mutant allele was suppressed with the exception of maternal T cells, where this was flipped.
You have 2 minutes.
I'll be done in 1. So this really suggested to us that monoallelic expression can correlate with disease. We did the same thing with PLC gamma 2, discordant phenotype between two cousins. One's B cells work fine, and the other ones they don't produce IgG. We sorted these cells, performed calcium flux, and here are the latest set of results. Wild type is on the x-axis, mutant is on the y-axis. Wild type individual expresses only the wild type allele, whether cells are fluxing or not. Individual who makes normal IgG by B cells, cells are fluxing or not, they are biallelically expressed. And the individual who doesn't make IgG, you can see the cells that are not fluxing are solely expressing the mutant allele. And I'll skip this one, but long story short, in the father who has the variation, we detect only the wild type allele. The kid that's sick, we, in classic monocytes and B cells, detect biallelic expression. In conclusion, genetic analysis can guide therapy in primary care setting. We have that. I think we're heavily underutilizing it. We need human variome project for the next century to come. We need to understand genetic penetrance and how genotype, and now a word we made up, transcriptotype, may differ because it may influence the disease penetrance. And a bunch of people who did this work and collaborators within and outside without whom this wouldn't be possible. Thank you.
All right, and great. We're going to hold questions to the end. So our next speaker is Scott Canna, Primary Immune Disorders and Specialized Infectious Testing.
I echoed Dusan. They turned it off. I echo Dusan's enthusiasm for this meeting. It's an honor to be here. I love how much time there is for argumentation/Q&A. Some disclosures that don't actually have any money behind them. You just heard from Dusan about, if you find certain infections or phenotypes, these are the immunodeficiencies to look for. And I was asked to go the opposite direction. If you have a patient with a known inborn error of immunity, or if I like primary immune disorder, because as you mentioned, not all of these are inborn, but it's going to take a long time to turn it around. What kind of infections should we look for? And so my next disclosure is, who is this guy? I'm a peds rheumatologist. My research focus is mostly on inflammation that's not infectious. And so either I'm here because you couldn't find anyone else, or because I represent this immune dysregulation program, which very briefly arose out of sort of clinical necessity because of too many argumentations over bedsides about what flavor of HLH someone might have. It's an inpatient and outpatient program. It almost functions like its own service. It has all these different specialists that you can see on the right, and is now sort of a institutional program of record. We're out of our sort of beta period, and serving as, I think, a little bit of a template for these around the country and how they might differ at different institutions. And so I drew upon my colleagues within what we call DIRT, Dysregulated Immunity Response Team, from the world of immunodeficiency and infectious disease, so thanks to them. Further biases are, we've talked about immunodeficiency as if it's its own thing, and I'm personally of the bias that there's really no such thing as immune dysregulation as a category. It's all immune dysregulation the whole way down, and that we shouldn't really be thinking of our patients as only having one mechanism of immune dysregulation. So finally, this is not a metagenomics talk. I'm hopeful that that's not why you asked me here, because I'm certainly not an expert. We have a little bit of that later today, hopefully. But I felt like it was worth at least discussing that there are multi-pathogen nucleic acid detection. Not all of these are metagenomic, but things you may have used or heard of. I'm sorry you can't see, but like BioFire or Carius testing is metagenomic, and these can be used on liquid samples like plasma or CSF, or increasingly even from tissue samples if you know beforehand that that is what you want to send. So I thought I'd illustrate the sort of idea of going from monogenic disease to infection with a case. So this is a 5-year-old with cartilage hair hypoplasia, which thank you, Dr. Puck, for introducing the importance of that gene as a potential inborn nerve immunity. And the child had that rash for quite a while, which was obviously very disturbing. And so has this defect in what is actually a long non-coding RNA that's important for this RNase, but doesn't really tell us a whole lot about how you get the phenotype. And so these patients have both a sort of syndromic appearance as well as a really heterogeneous and potentially severe immunologic phenotype that can include autoimmunity and immunodeficiency and something that I call sort of dyshematopoiesis, including bone marrow failure, but most patients don't have all of this. But this particular patient did have low T cell numbers and hypogammaglobulinemia, and was on replacement and had, to all appearances, seemed to do pretty well. But this type of a rash had really been there somewhat continuously through 18 months, or from 18 months. And so, of course, it was biopsied, and it showed sort of unremarkable non-caseating granulomas that had been cultured, that had special stains, that did not respond to empiric antimicrobacterial antibiotics. And so, what the heck is going on? Is this all just immune dysregulation? And it turns out that they went back and they found this is vaccine strain rubella. And this is all Kate Sullivan's story. So you can't read it, but it's down there. So if you take tissue blocks from these biopsies and you stain for rubella proteins, you can find either within simple or multifocal granulomas that this is a patient that has vaccine rubella reactivation that has caused this really hard to diagnose granuloma skin disease. And it turns out that about 70% of primary immunodeficient patients with granuloma skin disease, they find rubella. This is Kate Sullivan has grabbed slides from colleagues through USAIDnet and sent them off for special testing. It's actually done at the CDC. And so this is a breakdown of the kinds of immunodeficiencies associated with this sort of reactivation of rubella. Most of them had sort of combined immunodeficiencies, but you can see a decent number of just regular CVID. And so you can test for this. Now, if you have a suspicion for rubella, you might actually do rubella-specific testing, either with a targeted qPCR or through metagenomics. If you're looking through the retrospectoscope like they were, you can take FFPE slides and through a fairly onerous process send them to the CDC for testing. So that's just sort of an illustrative example of if you know that the patient has a T-cell immunodeficiency and has a granuloma skin disease, rather than going through that long diagnostic odyssey, you might seek out a specific infection like rubella. And so that is just emblematic, and I'll go through a couple of the infections that we tend to think of as being sort of hallmark of certain inborn errors of immunity. That's the list of EBV susceptibility genes. Some of them have their own sort of names of the syndromes. And testing for EBV is both serologic and through sort of targeted PCR. The one reason I bring it up here is that a number of institutions, including our own, actually do a little bit of a specialized test where we will sort peripheral blood cells into T cells, B cells, and NK cells, and do a qPCR from those, because these inborn errors of immunity, when they have severe EBV disease, it's still in the B cell compartment largely, but in EBV HLH, such as there is a garden variety, which is much more common in Asia for reasons that are not super well understood, it happens that actually infect T cells and NK cells. And so, the nature of the immune dysregulation might determine where that EBV is actually a reservoir. And that is not a test that is sort of widely available. Susceptibility to HPV is a common feature of a number of severe T cell defects, again, sort of listed in a table there. Clinically, it's not quite ready for prime time. We don't actually test for HPV. We just assume based sort of phenotypically that things look like HPV, and that's what it is. But it does turn out that there are specific types of HPV that are maybe more specific to certain immunodeficiencies. the gamma type being maybe more common in CXCR4 disease or WHIM syndrome, and maybe also in some other T-cell defects like DOC8 or GATA2, which is not a T-cell defect, I recognize. But the technology to do this is largely done through something called rolling circle PCR amplification, which a number of places are working on rolling out, but is not clinically available. Mucocutaneous candidiasis, there's a number of genes associated with loss of activity of IL-17 pathway. And, it usually goes the other way. If you have a patient with mucocutaneous candidiasis, you're looking for the gene. But in these cases, if you happen to know that these patients have the gene, you might be looking for it. Finding the candidiasis in mucocutaneous areas is not always so hard. The hard-to-grow strains are actually more associated with invasive candidiasis, which is actually not associated with these defects, but more neutrophil defects. And then finally, it is insane for me to be presenting on Mendelian susceptibility to mycobacterial disease at this meeting with some of the folks who are here, but nevertheless, you asked me to do it. So here I am, and that is the list. Mycobacterial detection is really hard. These are incredibly fastidious, slow-growing organisms that are not always tuberculosis. And so the current testing includes sort of a all-of-the-above approach, and we still are sometimes worried that we're missing an infectious cause of a very low abundance of mycobacterium in a patient that maybe they don't have an incredibly severe susceptibility, but if they're just not fully clearing it, that could result in a granulomatous disease. Maybe that'll be the next rubella story. And then finally, enterovirus is not one virus, it's a family of viruses that particularly associated with agamma-globulinemia, which is BTK deficiency. A substantial number of them can develop invasive enteroviral disease, especially in the CNS. And so there's a number of ways of detecting that, both by qPCR, if you know specifically what strains you're looking for. But if you have a patient that has a sort of the right encephalopathy phenotype within one of these inborn errors of immunity, you might look broadly for enterovirus within these tissues. And so then finally, what you might consider inborn errors of immunity that are not always what we consider immunologic diseases, like sickle cell, like cystic fibrosis. At least cystic fibrosis patients, a lot of the problems they deal with are largely infectious. And so I would consider it a sort of barrier or a structural immunodeficiency, at least in 2024. There's a long list of genes associated with both CF, largely because they have resistant bacteria that grow up because they get so many rounds of antibiotics. They also get non-tuberculous mycobacterium, but also CGD, chronic granulomatous disease. There's a number of hard to grow or anaerobic or fastidious bacteria that unless you know to look for them, you might very well miss them. And so I'm trying to move quickly. One of the slides didn't make it through, which was basically, so I'll just summarize it. There's not a lot of primary evidence for the use of these metagenomic or large multi-pathogen panels for the diagnosis of infectious disease within primary immunodeficiency. There's a bit of literature in specific syndromes like sepsis, and there's a bit more in HIV, which is maybe the closest sort of surrogate we have. And that data is basically shows that it changed management in a substantial number of the cases that are reported. So I bounced that just last night off of our special immunology clinic at CHOP, which is the clinic that manages children with HIV. And I said, well, are you starting to use metagenomic sequencing? And the answer is no. Some of the barriers to that are surely cost, both for inpatients and outpatients. There's a lot of gatekeeping to the access for those tests. But they also pointed out that the infections that the HIV patients get are the same infections that they were getting 10, 20 years ago that we have good sort of molecular tests for. And so they haven't felt like there has been an unmet need yet. But the corollary to that is they may not know what they're missing, and they might have patients that have indolent infections that they just haven't found because they haven't looked for everything. So in summary, there are a few situations where individual inborn errors of immunity would cause you to look for specific testing. I think rubella is maybe the best example of that, but I illustrated a few others. But really, the sense that I have from working with our immunologists and our infectious disease and our multidisciplinary group is that it's really classes of infection. If you have a immunodeficiency where there's substantial T cell defect, there's sort of a panel of T cell deficiency pathogens, and you're looking for all of them, and it's not always a one-for-one kind of thing. It's really, you're looking fairly broadly for susceptibility to those kinds of infections. Basically we try to aggressively seek out getting good samples and culturing broadly, and especially if you're worried about mycobacterial disease, culturing for a long time. And I think we're still figuring out what the role for metagenomic sequencing is. There's been substantial investments, both commercially and at large academic institutions, in making that available. We increasingly make use of it, but and the data, although somewhat biased because most of it is funded by companies that are selling these tests, show that it changes management in a substantial number of patients, but there's really not a pipeline for access to that kind of sort of expanded sequencing of pathogens, whether it's for inborn errors of immunity or whether it's for just garden variety patients or HIV patients. So with that, I want to thank the broad team of people who are tired of me bothering them about how we would sort of get to this question. And I'm happy to talk more about this program, which obviously I feel very strongly about, and plug that we have a symposium that is free. Folks are welcome to join. Thanks very much.
Thank you. Moving on, we're going to have Stephen Kingsmore from Rady speak about NICU and PICU sequencing and variants predisposing to sepsis.
Thanks, Terry. Great to be here. If you had met me in the late 80s or early 90s, I would have described myself as a cellular immunologist. My first R01 was from NIAID, and my first Nature paper was positionally cloning the Chiari-Kagashi gene. Bet a lot of you didn't know that. All right, so what are the current opportunities to save lives and avoid suffering in infectious diseases by whole genome sequencing? So for those of you who are immunologists and not genomicists, you might not be familiar with this literature, but there is now strong evidence that a genome sequence can save lives and avoid suffering in children with a very broad range of presentations. This is the literature, the evidence base as of last year. And the statistics speak for themselves. In fact, there has never been an intervention quite like the genome in critical illness. I really said that, and I believe it. When you dig into this, you'll find that there's not a single cohort study nor a single clinical trial which has looked at genome sequencing in infectious disease. It's entirely left out. So what is the evidence? Might there be any evidence that a genome could do the same thing in infectious disease? So 3 cases. As I said, there's no cohort studies, so I just went back through our records and picked out 3 informative clinical studies. And the first one gets to, should recurrent infection be a new indication for rapid diagnostic genome sequencing? So let's just run through this together. Baby girl is admitted to our intensive care unit for prematurity. She's small for gestational age, and she has multiple congenital anomalies. She's dysmorphic, but genetics can't come up with a unifying diagnosis. She is in hospital for a long time. She has surgical repair of her congenital anomalies, and she has, during that hospitalization, recurrent respiratory infections, both gram-positive and negative. And these are very severe, as evidenced by sequentially requiring being put on a ventilator, on ECMO, for cardiac and respiratory support. The family are asked on several occasions about palliative care being an option for this baby, but there was no etiologic diagnosis, and they held out hope that she would grow out of this. So finally, at 7 months of age, a genome was ordered. At that time, the baby was in another of these septic shock events and required both inotropes and ventilatory support. The diagnosis was Coffin-Cyrus syndrome, and this is indicative of a broad set of genetic diseases that are not typically regarded as being immune deficiencies, but if you do a search of OMIM under recurrent infection, you'll find there's 884 such diseases. This is one of those. Palliative care was started the day that the diagnosis was made, and she succumbed later that day. So rapid genome sequencing at birth would likely have allowed the family to have made a very different decision regarding their baby child. So this study would suggest, yes, we are missing out by not having recurrent infection being an indication for rapid genome sequencing. Okay, second study, or second case. This is a little baby boy who's admitted at 19 months of age to the PICU with septic shock, and the pictures show purpura fulminans, if you are not familiar with that. I'd never seen it before. His twin brother, and he was a monozygotic twin, so twin brother had died 1 day earlier at an outside hospital. Our hospital's located 11 miles from the border with Mexico. We have a lot of cases like this of families who try to get across the border to get admitted to our hospital. In this case, the twin brother had died on the other side of the border. The story was very similar in that we have a history of recurrent respiratory infections requiring courses of antibiotics. I don't have time to go through those findings, but for those of you who are immunologists, I know that you're just having a mental high there, and already you're thinking about what's the differential here. So let's get to it. Indeed, a genome is ordered, and it shows that he has X-linked A-gamma globulinemia, and most likely so did his monozygotic twin, explaining both of their presentations. But really interestingly, one thing we don't normally think about with a rapid diagnostic genome is that we get whatever else is in blood. When we purify blood for rapid genome sequencing, we don't like wash the cells, spin them down, stuff like that. We just take an EDTA blood tube or a blood spot and turn that into DNA and sequence it. And so here's something interesting. He was actually culture negative, if I go back, upon admission. Now, he was presumptively treated for Pseudomonas because of that skin rash, but he was culture negative. And in fact, what we found by aligning the reads that did not align to the human genome, we were able to show that he had Pseudomonas. And you can see the distribution of alignment of those reads to the genome in the graph on the right. He actually had a good outcome. Horrible hospital course, required debridement of those lesions followed by skin grafts, but he did well on IVIG and aggressive anti-pseudomonal antibiotics. But again, a rapid diagnostic genome done earlier in the clinical course could have avoided this hospitalization and the death of his twin. Okay, third case, even more interesting. We got it into our minds. Now, this is kind of before Carius came along and metagenomic analysis of cell-free DNA became something that's now evolving a standard of care for inpatient infectious disease workup. And here we just said, hey, let's take a look at unaligned reads in general. Let's align them across a host of pathogen genomes, and let's see what we find. And one of the findings that really jumped out to us was cytomegalovirus. And believe it or not, about 4% of 669 critically ill children who received rapid diagnostic genome sequencing had reads that were 100% aligned to cytomegalovirus, and the Q scores were such that, you know, they were high quality alignments. And for a subset of those newborns, there was sufficient clinical information to show that they were symptomatic for cytomegalovirus infection. And for some, we were able to show that there was a very nice correlation coefficient between the results that we found and qPCR. So this is really an unexplored area. We don't know the limits of this. So I've shown you a bacterial genome. I've shown you here a DNA viral genome. We have no idea how good a host genome is at metagenomic analysis. Okay, moving on then. Terry asked me to be very, very implementation-centric, and sometimes I listen to Terry. Make the most of it, girl. The policies, there are now this tapestry of policies, particularly for Medicaid across the country, related to rapid diagnostic genome sequencing. Each state that's colored here has its own policy, and none of them are common to two. And then on top of that, Anthem Blue Cross Blue Shield has policies which cover most states now, and again, their policies are different. These policies are complex, but the point I wanted to make was 2 points. One, it took cost-effectiveness studies to actually see that happen. Showing clinical utility in and of itself, that second slide I showed you with 44 studies did not get the job done. It was real-world evidence of cost-effectiveness and actually net cost savings that drove Medicaid policy directors to write policies for their states. Not all of those policies are effective. Some of them are wholly ineffective in as much as they provide no payment. Very interestingly and topically, and you're all going to love this, it is Republican states that are reimbursing rapid diagnostic genome sequencing, and Democratic-led legislatures are saying, we're not going to cover the cost of this test. But nevertheless, it's a policy. I won't go into that any further. Okay, so what might this clinical research and implementation study look like that we might want to do in infectious disease? Well, here's a study we published last week in American Journal of Human Genetics, which unfortunately was too late to get considered for one of the top 10 papers of the year. Darn it. Probably wouldn't have got in anyway. Here what we did was we said, let's only enroll babies. Was there a comment there?
He said
Next year.
Okay, I'll take you up on that. Here we said, let's only enroll babies for a rapid diagnostic genome who everybody says this kid is not genetic. They don't smell of genetics. They're not going to get a genome. So we enrolled 120 babies, and we ran diagnostic genomes in the pink on the right, and lo and behold, 30% of them had genetic diseases. I took a look at this on the plane up here yesterday, which was a bumpy ride, believe me. Anybody give me an amen to that? Thank you, Weather. Three of that 30% diagnostic rate were actually recurrent infection or diseases that are associated with immune deficiency. Again, these are not classical immune deficiency diseases, but they fall into this second group of genetic disorders for which recurrent infection is a prominent clinical feature. And just to point out the middle blue area there, which is one of the ending points, which is that there are now multiple groups worldwide enrolling lots of babies for a genome-based newborn screen. And there are hundreds of primary immune deficiency genes on those lists, since they are remarkably, just about all of them have effective therapies. So they're high on the list for addition to newborn screening. So last slide, So here are some hypotheses I'd like to throw out that we might want to test with grants. Hypothesis one, that rapid diagnostic genome sequencing actually does have sufficient clinical utility in infectious disease to warrant coverage policies, and I've thrown out some ideas there: severe sepsis, septic shock, sepsis with opportunistic etiologic agents, sepsis without a normal response to an adequate antibiotic regimen, TREC positives in the newborn screening area. Great opportunity here to do what I asked provocatively of Jennifer: could we do better than 3.5 months? And so if you marry the TREC positive with a rapid diagnostic genome, you can actually have that diagnosis in a week in terms of the molecular etiology of TREC positivity. Number two, just exploring this area of whether you get a free metagenome in a rapid diagnostic genome. You know, the alternative is to do something like a Carius test. There's no doubt that that's effective, but maybe we get a free analysis whenever we do a host genome, and very interesting to contrast and compare the two. This is just an unexplored area. Certainly for something like cytomegalovirus, we might also be wanting to add that to our newborn screening, as others have said here, and other congenital-ish infections that are probably massively underdiagnosed. As I mentioned in the newborn screening, we have 884 genetic diseases that we think we ought to be adding to genome-based newborn screening, since they are all associated with recurrent infections, and all of them have effective interventions. And then lastly, another unexplored area is getting outside of children's ICUs and getting outside of the NICU and saying, how applicable is this, say, in adult populations? Again, we have no idea. There's only one paper looking at the cardiac intensive care unit at rapid diagnostic genome sequencing. It found 20% positive findings in that population. It was only 50 subjects, and so it's insufficient really to understand what that might mean in terms of generalizability. Thank you.
All right, that was a really interesting set of presentations, so let's open for discussions. Down 80% down this table on the side, I can't read it.
Yeah, we may have to ask the people on this side of the table to kind of lean back a little bit so that our two moderators here can see. So there's a question way at the end near Bob.
Nope, just went down.
Oh.
Oh, so over here then.
George.
I really enjoyed all the presentations. Stephen, just a question for you. I was very impressed with the slide that shows the effective and ineffective strategies. We're certainly very, very interested in the clinical implementation and sustained use of effective interventions. But to what extent are the ineffective interventions also continuing to be done? Because another part of our interest in de-implementation of low-value strategies.
Yeah, so that slide was referring to policies, and that's my subjective opinion of whether it's effective or ineffective. There isn't published data on this yet. Most of those policies are just kicking in, and so there isn't historical data to judge this. But at least in our state, California, which was the first to have a Medicaid policy, so great, but there is no attendant reimbursement for the hospital. So they say it should be covered under the DRG, but as we all know, a hospitalization is only 97% covered by the DRG under California Medicaid anyway, so adding cost is not going to help. What we found is that there is no uptake, right, outside of 2 children's hospitals. Really, California remains not a state that has converted to a NICU regime where rapid diagnostic genome sequencing is done in a substantial proportion. We don't know the ideal proportion yet. There are some innovator hospitals like ours, like Mayo, like Riley Children's, where up to 1/2 of NICU admissions are receiving a diagnostic genome. They are showing cost effectiveness even at that scale. And so that's another question. So it'll take some time to sort out what the policies ought to look like, but a big gaping gap, as I mentioned, is there's no mention of infectious disease as an indication.
We're going to turn to Mark now, online.
Thank you. This is a question also for Stephen. The CMV data is really extraordinary, and I was wondering, and this relates to one of your extended research questions, as to whether or not you looked at other torch infections, rubella, toxoplasmosis, which would seem to be the most likely ones based on our understanding of their teratogenicity to look for.
We've not done that, Mark. I think it would be an exceptionally interesting question. The data's out there. I mean, it would not be expensive to do this on a large scale against a much broader array of pathogens.
Okay. Question over here. I believe it's Inka. Can't see your name tag.
Yes, thank you. So I think I have a question for Scott and both Stephen, or questions and comments. So some of the approaches that you showed, and I thought that was really, I didn't even think about, human whole genome sequencing and picking up pathogens in those samples as well. But I think, as was pointed out, the institutions that are doing the current sequencing, both for pathogens, metagenomic type approaches, are mostly academic institutions and maybe some hospitals that have a research arm in it. So the big question is, going to the sequencing first approach and figuring out what those patients have in terms of infection. I think there are also, issues around regulatory aspects and getting reimbursements for approved diagnostic tests, and we have a session on this later on. But what are your thoughts on that aspect of it, like around the regulation of some of those technologies? Either one.
Are you talking about the patient sequencing or the metagenomic sequencing, or both?
Well, I get the metagenomic for pathogens, I assume, and then I guess the same if you're picking up with the human genomic sequencing, the pathogens as well. But let's start with the pathogen metagenomic type approaches.
Yeah, I mean, I think that some of it is, I guess it's sort of the snowball is still at the top of the hill, at least for my institution in metagenomic sequencing. It's a thing that we talk about at a much higher proportion than we actually send. And then I think our accumulation of anecdotes of times that it's been useful is not high enough yet that we're sort of pushing. But I think that traditionally that's kind of the trajectory that these kinds of things have taken, is that the academic centers find it to be clinically useful and then have the resources to sort of fight for getting it done, and then push to, both from a research standpoint and from a logistics advocacy standpoint, get it adopted more widely. So I'd be interested in others' sort of clinical experience with the utility of metagenomic sequencing in practice, because I was surprised that even reaching out beyond our sort of group where we only deal with the really puzzling cases, that it's not caught on in some of our special immunology clinic or our transplant clinic.
Down at the end of the table on our side.
Yeah, I had a follow-up to that question. One of the things that I've seen in some of the guidance is a pretty strong distinction, so I guess this is probably also for Stephen and Scott, a pretty strong distinction between the inpatient setting and the outpatient setting, particularly for metagenomic tests and when to do it, with the one exception being among folks who are immunocompromised, where there are tests that would typically only be done in an inpatient setting, maybe, but for somebody who's immunocompromised or has a particular genetic condition, that maybe those metagenomic tests might have more utility in the outpatient setting. And so I wonder if you all think about that or have an opinion on that.
Well, certainly the emergency department comes to mind immediately, right? And could we speed it up enough so that we could predicate admission decisions on results? Wouldn't that be wonderful?
Yeah, I mean, I think that's a factor of the, like I said, I was not giving a metagenomic sequencing talk, but a lot of the data that support the use of those is exactly about sort of turnaround time, and that you can get results much quicker, especially for organisms like fungi or mycobacteria that take a while to culture, that I don't think has really factored into decisions to say, yeah, we could recover that organism by culture, but there's both value and I guess cost savings to having that answer anywhere from a couple days to a week sooner.
This is work. Good morning. Liliana Brown from NIID. We do pathogen genomics. Inca is in my group as well. I have a question for Dusan. I think I said it right. I don't know. But you had a wonderful presentation about RNA and how you can see different expression and how that can lead to different disease conditions. So my question is more using RNA sequencing for diagnostics rather than relying on genomics. It would also address some of the data that I saw in Stephen's slide where 2 reads are going to help you diagnose a disease, which would be incredibly difficult, I think. But in RNA-seq, you might be able to see more of that pathogen-seq. So just your comments on the reality.
Thank you for the question. And yes, that's how I was named Dusan, but I go by whatever is easiest. So in terms of host genome understanding of susceptibility to infectious diseases, I would go DNA first. And in the families where it doesn't make sense, or there is this form of incomplete penetrance, which is actually way more common than we like to admit, or we report in literature because it's mostly shrug, we don't know what to do. Absolutely, we need to go on the RNA level because we, and hopefully others as well, are documented that this discordance can happen. And because it is RNA, often that allows for sequencing of not just the host, but something else. So it can be a catch-all moment if designed appropriately. Of course, it gets very complicated very fast because we are talking about somatic commitment to transcriptional of a one allele. Then that means it's in a specific lineage. Let's be simple and just talk about the immune system now. Specific lineage of the immune system. So I think we are a few years away of being able to do this well on a single cell level, where we can then pseudobulk the parts and lineage trace based on mitochondrial DNA burden and infer what is this lineage that may be more committing to maternal or paternal. So I think it's still early days there, but absolutely going forward, I think we should be developing it.
We're going to have Jeff and then Jennifer.
Thank you. Stephen, always great to see your data. I'm wondering if you've had any, Stephen, if you've had any experience or success in working collaboratively with a payer or payers to design your clinical trials so that you agree on the endpoints, and if those endpoints are achieved, then a coverage decision is likely to be made in favor if the data is supportive of that? Or is that a completely naive way of thinking about it?
No, it's not at all, Jeff. You almost never say things that are naive. With Anthem Blue Cross Blue Shield in California in particular, we worked for about a year with their medical director, who was very open to suggestions in terms of what a policy might look like and where there were gaps in evidence. And having established that relationship, he then went to bat to get a national policy across all of the blues, and has continued to be in touch with us to look at modifications, knowing that a first policy document is not going to be perfection, and where should it be tightened up. So that was an exceptionally good interaction. More recently now, we found that the same is true of Medicaid medical directors, and that this is a group of people who really are looking for hard scientific evidence. You know, they really read the literature, they sweat the details, they produce these 100-page long synopses of all the published literature, and the onus is on us to publish well and have well-designed studies that they can really draw conclusions from. and then to reach out to them. And we've seen this now happening state by state, where we now just routinely will reach out, as legislators get interested in their state adopting something. We'll typically have an outreach, and having government relations folks seems to be a good way to do this, that they know the legislators and are able to broker that discussion and then get passed along to Medicaid medical directors.
Jennifer?
Yeah, this may be for Scott or perhaps Stephen, but in patients with immune deficiencies, getting a tissue diagnosis is absolutely critical, and yet we in actual clinical practice, we know people want to use the available sample or and so we find a lot of the time people are getting a bronchial lavage aspirate when we know the yield from that is dismal compared to actual tissue. So I wonder, the NICU whole genome sequencing is always blood. Have you thought about other kinds of samples that might have better yield depending on a phenotype? And I think that is something that, especially for immune deficient patients, you should think about up front.
It is dogma that we feel like, that we get much better yield out of tissue. And I wonder if a lot of that is because our diagnostic methods are maybe more antibody-based, because I was impressed as I was looking through some of the metagenomic literature that at least in HIV, the diagnosis of PJP from BAL and paired blood was pretty much 100% concordance. And much, much better than silver stain or other, more traditional PJP methods. So I think that, using more sensitive techniques that are looking either for specific pathogens or more broadly at nucleic acids might overcome some of that dogma that, that might be where the investment needs to be because it's probably easier and cheaper than trying to revisit, going back into tissue blocks and doing these things.
I think we need a lot more research in this area, Jennifer, honestly. We don't know the kinetics of DNA emia, really, right? And particularly cell-associated DNA emia. The beautiful thing is you're not relying on culture, so you can have dead bugs and you're still going to pick them up. That's awesome. We know that we have exquisite sensitivity. We can pick up a single molecule, and we can have a bar in terms of its match level, so that we're certain, even with very few counts of positivity, that we're seeing something that's not an epiphenomenon. So I feel good about that, but honestly, we just need a bunch of research to understand what are the kinetics, what is the sensitivity, how does that vary based on the type of etiologic agent and the site of infection, right? DNA emia is going to depend a lot on things like, are there barriers to, DNA getting into the bloodstream? It's a great question.
Next, we'll go to Ivan and then down to Bob Nussbaum.
All right, thanks. So my question, and maybe this is not just for the speakers, but for the people in the room. So I love this meeting because it gets together genomicists, immunologists, and infectious disease specialists. And so, at our institution, the gatekeepers, as Scott mentioned, to getting cell-free metagenomic sequencing are the infectious disease specialists. And, I guess the question is, what do we have to do, or what should be done to maybe get them to open the doors a little bit more for this kind of testing in immune deficiency patients? We have so many patients with immune deficiency, genetically diagnosed, who come in with fevers, signs of infection. You swear there's an infection there somewhere. You can't figure out where it is. We want to send this test. They say no. And yet they're also get this, the antibiotic stewards of the hospital. So, after 2 days, they're like, stop all antibiotics. I mean, perhaps the most frustrating disease, and Steve Holland will appreciate this, is CGD, where, I mean, they're sick. They have all signs of infection. We cannot find the bug, and we don't know what to treat them with. So, like, what can be done, or what do we need to do? And like Scott alluded to, there are no studies, and this is what they tell us. There are no studies that show that doing this test has any kind of benefit in immune deficient patients. So maybe that's where to start.
I mean, that would make the absence of evidence argument, like, there are no studies. That doesn't mean that it's not true. It just means that, like, we have to do the studies. But, I have this argument with my ID people, and like, just because we haven't done the study to show that it's true doesn't mean that it's not true. And so,
So it might be helpful later on for you all to define what kind of studies you're thinking of, okay? I mean, because we're kind of talking about, well, we need more research on a variety of things, but if, think about this over dinner or whatever, over lunch, what kind of studies are you thinking?
You guys are just ganging up because Jeannie left the room. I know that. But I really want to echo that. I think as they say, the absence of evidence is not evidence of absence. I would just put one caution in there. The whole reason to do genetic diagnostics is because we think every disease is different. And I think it is true that every immunodeficiency is a little different. And there are the ability of cell-free DNA diagnostics to identify pathogens in chronic granulomatous disease is markedly inferior to the ability to do it in other, say, T-cell defects, for a whole variety of reasons. And we need to generate that database, and it is in part incumbent on those of us who are doing it to put it together. It'd be great if the companies that have an interest in this created studies. And I'm not trying to defend my ID colleagues necessarily, but they are correct that we don't know. And that is really an urgent call for us to get the hell out there and find out. And whether we have to do it by pooling clinically relevant data or setting up prospective studies, I think is an issue for us to discuss.
I really like that study. I mean, I did the study, so of course I like it. Where we only enrolled babies who everybody said, no, that's not genetic, right? They're not going to get a genome. Doing something similar with metagenomics would seem to be the way to go, right? That's quite humbling for the gatekeepers, right? When they realize that their paradigm and their filtering tools are no better than tossing a coin, right? And it may or may not be true. It may be that they're great at calling who should get that test or shouldn't. It's a great way to do it. We have the same phenomenon with genetic testing, with host genomes, where the typical gatekeeper is the medical geneticist. And again, it's been sobering and life-changing for them when they realize, that was Coffin Cyrus. Why didn't I pick that up, I had 8 months to look at that baby, and I didn't think it was, So I think we're going up this learning curve, which comes from genome first, metagenome first, where we are relearning as physicians the fact that clinical presentations are much broader than the textbooks tell us.
All right, so let me go to Bob Nussbaum, who's next question.
This is actually for Dusan. I wanted some more clarification. So first, it looks like some of the monoallelic expression looks like it's parental origin imprinting, but there are others where there are multiple siblings who inherit the same variant from the same parent, and yet they vary in their expression. And I'm wondering if he has any insight into what the mechanism is for this. And my second question is, for the ones that show parental origin effects, are those genes sitting in regions that have been suspected of being imprinted? And are there neighboring genes that also show monoallelic expression?
Right, so it appears that imprinting must be the whole organism, so parent of origin. Whereas in the random autosomal monoallelic expression, none of the genes lie in imprinted gene regions. They are, by definition, must happen somatically, otherwise they're not random autosomal molecularly called, because then they are parent of origin. And they are seemingly stable, and unlike imprinting, they're not 100% mother or father. They can be, the percentages that we came up with is above 80% single parent origin expression levels. We've done the calculations for 90, 95, but it's also true at 55, 65, and 75. And it's a completely arbitrary point. And where that cutoff should be used in terms of known genetic causes of diseases and penetrance is probably a sliding scale.
So if I could just follow up, so do these represent then somatic errors of epigenomic effects around these genes? Does the same gene always show some monoallelic effects?
No, it doesn't. So yes, these are de facto, by definition, somatic epigenetic errors, if you think, because they're not biallelic, which is what biology should be. What we do know is that maybe it's not an error, because healthy New Yorkers that we tested, about 0.5% of testable genes in any individual can undergo it. Collectively, it's about 5%, and it's not always the same gene across, but there is a propensity of certain genes. But we're still not at a saturation to understand if it's a specific class. They're not organized genomically in any fashion that we or others can understand, so TBD.
Next to Inka. I'm sorry, I don't know your name, sir. Yes, thank you.
Thank you. Circling back to the question that was asked before about the clinical value and the gatekeeper question. One thing that'll definitely help is when the test is not $2,000. So that's a barrier, which will come down in time. Yeah, sure. It's just commenting on the fact that one thing that'll help is when the metagenomic test doesn't cost $2,000. And that's something that should come down, obviously, over time. But one of the other challenges is demonstrating the incremental value. And there are some papers that have done that more so in an adult immunocompromised population, so maybe not necessarily the neonatal population with inborn errors of immunity, for example, but in transplant patients, severely immunocompromised patients, the data is there to demonstrate that there is incremental value. But you have to end up restricting it to those who are really highly likely to have atypical organisms that you're unlikely to find using conventional methods. And it's also worth noting that it's not a good enough test to replace other testing. So for example, there's a partial overlap with what you get from culture. There are things that metagenomics misses that end up growing in culture and other methods. So it doesn't end up becoming a replacement. It's an add-on, and that just further challenges the economics of it.
Terry?
Yes, so I wondered if some of the genomicists who know this better than I in the room could speak to Dusan's question about a human variome project. There is such an animal from the HUGO, the Human Genome Organization, I think it is. And we also have the impact of genomic variation on function, the IGVF program that's addressing it. But Jonathan, maybe you could speak to that, or I saw you nodding, so, but maybe someone, or Bob, someone else.
I don't know the details other than to say I think you're right, that there are already some nascent projects that could be sort of tapped into for that kind of idea. Yeah.
Yeah, it might be interesting, Dusan, to take a look at that project in particular, and also the IGVF, and see where you're seeing holes or gaps in what's available there.
I would put forth, what I heard you discuss was what I perceived as since you made it analogous to the Human Genome Project, hit the accelerator and try to be comprehensive. I don't believe, our IGVF program, Impact Genomic Variation of Function, I think we should still regard as a exploratory pilot, figure out what we're going to do phase. I don't think we believe it's ready to fully hit the accelerator, and we would fully hit the accelerator would be bigger than our institute could do. So I think we are, as an institute, that's very high on our priority list to try to lead an exploratory way, because we know that's an incredibly important area of genomics that fits very much in the wheelhouse of what our institute should be doing. But I will acknowledge it's not probably at the scope and scale of what your enthusiasm sort of implied. But it's something we're certainly hoping to help figure out the path forward, and also develop the technologies, the computational needs, and the strategies for eventually hitting the accelerator harder.
If I can just say something, is Liliana again. If there is going to be a project studying the variation of human genome, I would urge you to call it something else than a variome, because we already started the human virome project, and at least the word would be very close and confusing. But also just a note to mention, for example, for SARS-CoV-2 at NIID, we've done a lot of work studying just within the receptor binding domain the implications of variations in a very small region of that gene. And it's a lot of work from a lot of many different groups. And that's in one single pathogen in a very small region of a protein. So it's not as easy as simply annotating a specific mutation. It takes a lot of work, and yeah. Anyway, I'll stop there.
Dan, online?
Yeah, so I'm online. I'm at home with coughing and sneezing. Terry thinks I have pertussis because she's seen a case, and I'm not going to argue with her. Maybe I do. Sorry, you don't want me there. We have spent a lot of time, we're spending a lot of time developing assays of variant effect for, dare I say it in this meeting, cardiovascular genes. And one of the things we're learning is that every assay assays a specific protein function. So, when we start to look across literally tens of thousands of variants in our favorite genes, what we find is we can assay specific protein functions, but then we have to take one step back and say, well, did we capture everything that's pathogenic or benign? And that's going to be an Achilles heel to the variome project, or whatever it is you want to call it. The other two things to say about that approach are that eventually we'll have enough information that we should be able to use AI approaches to infer the rest of function. And it's the marriage of AI, structural biology, and actual deep function at the level of thousands or tens of thousands of variants is going to be the next big frontier for this. And I'm glad to hear Eric's enthusiasm. We're equally enthusiastic.
Bob? Who's ever next to Bob at the end of our table? Sorry.
Yeah, I have a question, I think for Dusan, regarding the monoallelic gene expression. Did you look at differences in DNA methylation that were allele specific, and did that play a role at all?
We did. So DNA methylation does play a role, as does H3K27 trimethylation, and we've also looked at H3K4 methylation, and the first two play a role, but it's actually more complicated than I thought, isn't it always? Even in the clones from the same individual that have committed to alternate or reference allele, in some clones, when we downregulate DNA methylation, we can actually now switch back to biallelic expression, right? So that's evidence that DNA methylation plays a role. But when we do the same thing in a different clone from the same individual that's at the alternate allele, nothing happens. So it seems like these marks are necessary but not sufficient, and sometimes they suffice. So it appears to be a fairly complex epigenetic regulation and decently leaky, because the level of monoallelic expression, unlike in imprinting where DNA methylation just shuts it off completely, right? So you just don't have it. Here, it can really be, 50 shades of gray from 50 to 100.
So I'm wondering if we could ask the three speakers from this session. We had asked each of you to focus on sort of one thing, whether it was pathogen-first, phenotype-first, or sequence-first. Some of you followed instructions, some of you didn't. But no, at any rate, and obviously, the answer is yes, we want to do them all. But are there ways of identifying, when is it the pathogen that really points you toward immune deficiency? When is it the phenotype, and when is it the sequence? So maybe you could comment on those, please.
Can I start? So I still consider myself relatively young, which is wrong, but I host that belief. Thank you for that. But part of that is because I'm very privileged to sort of be surrounded by, the really paramount leaders of multiple fields who, like Dr. Puck, who have been seeing patients as the whole field has evolved. And the refrain that I hear from a number of them is that despite the exponential increase, we're still lousy at guessing what gene or even genes it's going to be. Every time. Thank you. So, and it's not because, these are some of the most brilliant people I've ever worked with, right? So, and they're paying attention the whole way. And so I think that, part of this is just that the phenotypic expression of inborn errors of immunity is very incompletely described, in part because that's work that no one wants to fund. It's not necessarily a hypothesis-based thing. It's, we have to build databases and populate them with phenotypes, and that's not it's hard work that costs a lot that nobody wants to fund. But I think part is just that these things are really pleomorphic, and so I think that favors, for me at least, a very genotype-first approach, because we have not iteratively gotten better at predicting what the genotype is going to be.
So I'll agree with Scott here. It's very hard for me to understand any reason that when a child is in NICU or PICU, doesn't that get whole exome or whole genome done. I think that should be a standard of practice at the price point that we have right now. It's just the first thing that gets done in parallel to everything else. And I even think bigger than that, but in these cases, and in adult setting, I would go as well, especially with the explosion of somatic variations that have adult onset as well. We absolutely should be doing that, because it's still the very minority of all of us who will ever end up in ICU of any sort, right? Which means something is very wrong with us.
So to me, genome first means that we are doing screening, and phenotype first means we're doing diagnostics. And I think we need both. I think we need a belt and braces approach. I think they both have different pretest probabilities of positives, and so the test formatting is completely different, as is the cost structure, the size of the population to be tested. I think there's a strong case to be made for both.
All right, I'm getting the nod from Terry that I think we can we wrap the session, or are there final comments?
If there are other comments, I mean, we have all the way till 12:35.
Can I ask a question?
Yep.
So we were just talking about how there's a big gulf between what the cost is for research sequencing versus, the clinical test, right? So I just sent out a trio whole genome to be sequenced at the Broad, and it cost a little over $1,000 for all three. And that is continually coming down, yet, it's impossible to know what anything costs clinically, because there's what's charged and what's paid, but it seems to be in the many thousands. And I understand that there's a lot that happens, isolation of DNA, and then the big bioinformatics that must happen, although, to be honest, that looks like a lot of that is just sort of automated. And so I'm just wondering, what are the barriers to shrinking the cost of clinical sequencing when the cost of the actual sequencing part is so precipitously dropping?
Oh, I can go with that one because I live that nightmare day in, day out, every day. So we need automation, right? Clinical genome sequencing is incredibly manpower intensive. And we've talked about the VUS issue, and we're talking dozens of hours with incredibly highly paid people and 3 analysts to interpret a genome. And we're still only, diving into the top of the surface. If you pay for a $1,000 genome, you get $1,000 value. You pay for an $8,000 genome, you get $8,000 value in terms of the extent, the thoroughness of the interpretation. How we fix that, honestly, is automating interpretation. We're not that far off from it. There are now really good algorithms that are about 95% good, right? So as a first pass, if you want a $1,000 genome, these tools are awesome. That's kind of how the Broad is doing it. So it will come down. Part of this is it's still an esoteric test, way underutilized. Enough on that. I could go on all afternoon.
If I could weigh in on that as well. A lot of this is the confusion, and not to say that the questioner had that confusion, because I think he clearly didn't, but we conflate cost and charge. And what it actually costs to do something and what we charge for that same thing has a lot of different components to it. And one of the things that we've encountered at the institutional level is that with send-out laboratories, many times the institution that's sending out the test applies a multiplier for charge, which if it's a relatively low-cost test is no big deal, but if it's a test that costs $1,000, then that adds up quickly. And that, of course, has to do with how we do insurance reimbursement and the fact that different insurers pay different amounts based on charge. So it gets incredibly complex to deal with the charge issue and what is relevant to the patient and ultimately the clinician who's going to have to deal with the patient. And those are not amenable to sort of generalizable solutions for the most part. And so that's more of a policy issue than it is something that's intrinsic to the actual testing process itself. And it's very frustrating.
I might also add to that the difference between inpatient versus outpatient testing and way those things get funded in healthcare. So if you're doing an inpatient genome, the hospital that's doing that test is probably going to pay directly to the sequencing vendor, the commercial vendor. And that's going to be a small drop in the bucket if you're a NICU patient and you're there for 3 or 4 months, right? And so from Stephen's, his group has really sort of pioneered this and shown cost of the genome, according to the cost of the NICU stay, it's a no-brainer. And most institutions can see that logic and start to think about how they can just pay for the genome because it's going to save them money. As an outpatient test, it's getting billed to insurance, and that insurance might be a private insurer that will or will not cover under certain circumstances that test, and it might be a public Medicare where there's this patchwork of decision making. So it just gets really complex at the level of outpatient testing. Who's going to pay for it? I would also wager that the Broad Institute's probably getting a much different deal from the sequencing companies on reagents and machines than other groups do. So there's a lot of subsidy that's happening behind the scenes probably that also is affecting what's the apparent cost that you might see as the investigator.
Could I suggest, I know we're just before lunch, but we have 2 people at the end of the table who have a lot of experience in this, Bob and Jillian. Could we maybe take those 2, Lucas and Jonathan, and then we'll stop? Okay.
I was just going to add an administrative detail to Mark's to call out that from a cost and pricing perspective, many of the prices for tests in this country are based on what CMS sets the rates for. And for exome, there is what I'm pretty convinced is a typo in the fee schedule that leads to the cost of an exome being $30,000 per the CMS rate, and specifically the comparator code. So if you do a trio, that is priced at $12,000 per comparator genome for parent. It's a little bit better for genome. It's about $15,000 is what our government has priced genome at. So I think that's also something that somebody could look at if they wanted to reduce the cost of exome and genome.
I just wanted to add one other area that adds significantly to the cost is the billing department. The billing department for example, at Invitae, I don't know the exact numbers, but it's comparable to the size of the entire variant interpretation group, in fact, even larger. And between having to do pre-auths and letters of medical necessity and appeals adds tremendously to the cost of trying to deliver these results.
Okay, we'll wind up? Great. Before we applaud all of our speakers and presenters, lunch is outside. It's a little bit sticky because people who are paid to attend the meeting, your lunch is already paid for. You should have gotten an email if you're not paid to attend the meeting that you could pay for lunch, and you had to do that in advance. So if you're not sure if you've paid for lunch or not, you probably shouldn't eat lunch, at least not here. But at any rate, perhaps Jenna Cohen can help you with that. We will be back here at, I don't have it right now, 1:20 for our next session, and I think that will do it. So many thanks to all the speakers and presenters.